Hello everyone, welcome to today's seminar. So I have two things to say in advance. So first, this seminar will be recorded. And the second is you can unmute yourself and ask questions anytime, or you can use the chat box. Today, we invited my colleague Lu Ju from Dr. Zuo's lab. And here I will have a brief introduction. Ju graduated from Tsinghua University, actually with a major of engineering. And then he switched to neuroscience and got his PhD at Harvard University, where he studied the neuromuscular junction in Jeff Lisman's lab. Then he joined Mark Snitzer's lab at Stanford for the postdoctoral research. Then he joined our lab and focused on the calcium imaging. So today he's also have a lot of calcium imaging and further data analysis. Today he will share his recent work. He's collaborate with another PhD student in the lab about how the stress affect the brain circuit and how does the psychedelic drugs can restore everything. So let's welcome him. Go ahead, Ju.
Hello, everyone. So first of all, I would like to thank Xiaorong and other organizers of this online forum for giving me this opportunity to share with you some of my recent work. So as we all know, stress is a prevalent problem in modern life. So it may come from, as we all know, this pandemic and the ensuing social isolation, or it may come from like financial difficulties, family crisis, or work pressure. It may also come from more disastrous things like a natural disaster or political turmoil. But anyway, all these different forms of stress cumulatively can be really deleterious to both the mental and physical health of the person. So actually stress is one of the major risk factors for psychiatric disorders such as depression, anxiety, and even schizophrenia. It may also trigger problems in the cardiovascular system like hypertension and as well as diabetes. However, there is currently really not that much therapeutic approaches we can take to combat stress, either to prevent it or to revert the deleterious effects. And in order to understand the neurobiological basis of stress-induced mental problems and to find out the potential treatment, we adopted a rodent model of stress called the unpredictable mild stress. So in this paradigm, we basically subject the mouse to different kind of random mild stressors. For example, we may expose it to light in the at night, so the circadian rhythm is kind of disrupted. Or we may put it into a tilted cage, or temporarily restrict food access, or put it into tube for confinement, or subjected to kind of exposure to stranger animals or sudden loud noise and so on. So basically these stressors come as quite unpredictable and randomly presented. So they mimic this unpredictability and stochasticity of the real life stress. And in order to understand how the brain changes in response to these kind of stresses, it is, I think, necessary to remind you that the brain itself is a complex system with multiple organizational levels. So if we take the mouse brain and dissect it, there are a lot of different regions, each with a distinct function and connectivity with other regions. And some of them, like the ventral tegmental area, or VTA, amygdala, the nucleus accumbens, the prefrontal cortex, and hippocampus, are better studied in the context of stress. Other regions, such as the somatosensory cortex, so far have received less attention in the stress field. And actually the primary sensory cortex is the focus of our study. If you zoom into the sensory cortex and just take a slice of that, you will find like the cortex is organized in 6 layers with different types of cells distributed in these layers, and they in turn have their distinct morphologies and functions. And if we really zoom in to the to this intricate neural network, we can finally see the dendrites and the axons. Here, these are the axonal boutons, which are the presynaptic sites of synapses. Those are the connections that's between the different neurons, and they transmit information from one neuron to another neuron. And the green ones here, these guys show the dendritic spines. They are the recipient sites of the synapses. And the dendritic spines are one of the main objects we study in this project. So our work, excuse me. Sorry, I couldn't hear you clearly. Hello? Is there a question?
Maybe it's not a question.
No, okay. So in this study, we mainly focus on the synaptic level as well as the population of neurons. And previous work has indicated that the stress really disrupts the synaptic circuit in the cortex. So here is shown in cartoon is illustration of layer 2, 3, or layer 5 pyramidal neurons in the prefrontal area, cortex. And as you can see, after stress, the dendritic arbors get less complex. Some of the branches retract. And in addition, if you zoom into one branch, you'll see like a lot of the spines are lost. So you have a decrease in the dendritic spine density. And moreover, people have found that the receptors, the NMDA and AMPA receptors of the synaptic sites also decrease in their density, as well as other synaptic proteins get decreased. So how can we combat all these kind of problems induced by stress? And one potential approach is to use this class of drugs so-called the psychedelics. The word psychedelic is derived from 2 Greek words, psyche meaning soul, and dailon means to reveal. And because these drugs have like really strong and very fascinating effects on the human mind, so they can alter human perception and mood, and they can affect the cognitive processes. And a common feature of many of these psychedelic drugs, such as the LSD, psilocybin, or DMT, is that they are hallucinogens. They cause like hallucinations in the human. And primarily, such hallucinogenic effects rely on their action on the serotonin receptors in the brain. And over the years, people have tried to use such drugs to treat a variety of psychiatric disorders, including addiction, anxiety, depression, and some other anecdotal evidence suggesting that they may even be used to treat things like obsessive compulsive disorder. However, because these drugs are hallucinogenic, it is a big concern for clinical use. And that has really held back the studies on them because most of these are like Schedule 1 drugs, so they're very tightly regulated. So is there a way to get the benefits, the therapeutic benefits of psychedelics, but remove their hallucinogenic effects? And one attempt made by our collaborators in UC Davis, in Dr. David Olsen's lab, is to develop some novel non-hallucinogenic analogs of the psychedelic. And one drug they synthesized is called the tabernanthalog. Tabernanthalog is the analog of this drug called ibogaine. And the ibogaine is extracted from this plant, this shrub in West Africa. It's called the tabernanthe iboga. So ibogaine is hallucinogenic, and actually it is also toxic to the heart. But Dr. Olsen's group did a lot of function-oriented synthesis of different analogs and screening, and they found this drug, tabernanthalog, or we'll call it TBG, with several desirable properties. So first, it is soluble. So that's a kind of important feature for therapeutics. And the synthesis is easy. It is not hallucinogenic. And I have to mention that they haven't tested it on humans. So it is inferred by this so-called head twitch response in rodents. So some previous works have shown that all the classical serotonergic psychedelics would induce this rapid head twitch response in rodents. And that seems to be highly correlated with their hallucinogenicity in humans. And TBG has low cardiotoxicity, so that addresses the other problem of ibogaine. And finally, they found that it promotes dendritic growth in vitro. And we also did some experiments to show that this drug can promote dendritic spine formation in wild type animals. So then this drug seems to be a quite promising candidate to treat stress-related deficits. And first, we evaluated its effect on several behaviors of stressed mouse. So we started to stress the animal with this unpredictable mild stress paradigm for 7 days. And at the end of the stress paradigm, we give it 1 shot of low dose of TBG, that's 10 milligrams per kilogram. And we image, although sorry, not image here. And then we wait for a day and do the behavior assessment. So we use the 3 behavior paradigms. First is the elevated plasmase to assess anxiety. And then the whisker-dependent texture discrimination task to assess sensory processing. And the 4-choice odor discrimination reversal test for cognitive flexibility. And I will go through them one by one in the next few slides. So first, we did the elevated plasmase. So for those of you who are not familiar with this test, the EPM is a very popular model to assess anxiety in rodents. So the arena is elevated from the ground by about like ½ meter, and it has a pair of open arms which are just extending out, and then a pair of closed arms with walls enclosing them. And it leverages this conflict between innate drive for the rodent to explore versus its fear of height. And the interpretation is that if the rodent is in an anxiety-like state, it will tend to explore the open arms less. So as we can see here, the total amount of running in this elevated plasmase between the control, the stressed, and the TBG treated animals is not significantly different. However, the stressed animals tend to spend significantly less time in the open arms. So that suggests that they are exhibiting an anxiety-like behavior. And with TBG treatment, this deficit was completely rescued. The next test is this 4-choice odor discrimination and reversal test, or just 4-choice test. So here we have arena divided into 4 quadrants. And in each quadrant, we can place a container, basically a ramekin, that is associated with a distinct odor. So the test comes in two phases. So in the initial discrimination phase, only one of these four ramekins is associated with odor that gets reward. And the reward is basically a small piece of breakfast cereal buried underneath the wood shavings. So the mouse cannot see it, it cannot smell the food directly. It has to learn this association between the particular odor and the food reward. So the mouse would just wander around and then spontaneously start to dig into one of these ramekins, and it may get a reward, or it may not get the reward. And when it makes the correct choice in 8 out of the 10 consecutive trials, then we consider it as having passed this discrimination test. So as you can see, the number of trials to criteria in all these 3 groups are not significantly different. However, in the second phase of the test, the so-called reversal phase, we change this odor reward contingency. So the previously rewarded odor is now no longer rewarded. And one of the previously unrewarded odor now becomes rewarded. So the mouse has to learn this new association and basically resist the temptation to follow the old association. And here comes the difference, right? The stressed animals perform significantly worse in the second phase of the task. So suggesting that it is much harder for them to change this association, or they are exhibiting a kind of inflexible cognitive processing. And after TBG treatment, this deficit is rescued, and TBG treated mouse perform as well as the control animals. And finally, we assess the sensory processing in texture discrimination. In this test, we first give the mouse the same texture on both rods. And the mouse has 5 minutes to explore this arena and to interact with the textures. You can see it in the video. Here it's actually for the second phase. So it would explore these posts and go around and then come back again. And because these two, in the first phase, these 2 posts are identical, so the mouse would spend roughly equal amount of time and making roughly equal number of visits to both posts. Then after a 5-minute rest, the 2 posts were changed. So one would retain this habituated or the familiar texture, and the other one would be replaced with a new texture. So these posts were wrapped with sandpapers, and the new texture would be just sandpapers with a different coarseness. So initially, as we can see, there's really no preference to either side. Right? So in the encoding phase, there's no discrimination index, which is defined by the difference of the number of visits to the 2 posts divided by total number of visits. But in the second phase, in control animals, they have this innate preference for novelty. So they would visit the novel side more. As you can see here, this higher discrimination index preferring the novel. But in the stressed animals, they basically on average have no preference for either one. Right? So they basically lost this apparent preference for novelty. But in the TBG treated animals, the preference is restored. And
can I ask you a question? I'm Yi Chang Jia from Tsinghua University.
Hi.
So this paradigm looks to me is very similar to lower object recognition. So is that lower object recognition as well? We call it.
I didn't do that novel object recognition test, but another postdoc in our lab who previously used a different stress paradigm, the 7-day restraint stress, actually checked that because he was also testing this texture discrimination in the animals, and he also did a novel object recognition based on color. And in that test, it seems like the animal behaved fine after the 7-day restraint stress.
Okay, I got you. It's something not object, it's textual. Is that right? Is that right?
Yes,
okay.
Yes, that's our interpretation. Yeah, and this texture discrimination is actually the basis of a lot of the later studies that I will talk about today. So having done these behavioral tests and found that the TBG has the rescuing effects on all of them, and then we went on to study the neurobiological basis. And I was using 2-photon fluorescence microscopy to study both the structural changes and the functional changes in the mouse brain. And just as a reminder, the advantage of the 2-photon fluorescence microscopy derives from the fact that it uses long wavelengths, actually infrared light, to excite the fluorophores. And it actually requires 2 photons to be absorbed simultaneously by the fluorescent molecule to emit fluorescence. And because of the long wavelength, the 2-photon excitation beam can penetrate into the brain much deeper than the 1-photon excitation do. And furthermore, because it requires 2 photons to be absorbed at the same time for the excitation, you have this so-called optical sectioning ability. So as shown here, if you just have 1 photon excitation of this solution that's I think fluorescent, you have this hourglass-shaped light, right? And everything in this bright air volume is emitting fluorescence. So you really can't decide where the fluorescence comes from if you are looking just from this side. But with 2-photon excitation, fluorescent emission is confined to a very small region around the focus, because when you move away from the focal plane, light intensity goes down, and because it's 2-photon effect, your emission goes down as the square of the distance. So it's essentially it is confined basically to the focal plane. And that is very desirable because then we can really see the 3D structures in the living brain. And in order to access the neurons in the mouse cortex, we can either do a thin skull approach, in which a small region of the skull, the overlaying skull, is very carefully thinned by dental drill to the point there's only 20 to 30 microns thickness left. And from that, you can actually image through it and see these are the dendrites on the superficial dendrites in the mouse brain. By the way, this image was taken from a Phi 1 YFP H-line mouse in which sparse subset of layer 5 pyramidal neurons express a very high concentration of YFP in their cytoplasm. Alternatively, you can do cranial window. So basically you remove a piece of the skull and replace that with glass cover slip and seal it around. So you can image through the glass window and to see the neurons in the brain. And here I'm showing you an example of calcium imaging. So these neurons are labeled by adeno-associated virus, or AAV, that encode a calcium indicator, the GCAMP6F. So as you can see here, some neurons increase their brightness, right? And that is because when the neurons are active, there will be calcium influx. And when the calcium indicator proteins bind to the calcium, they will greatly increase their fluorescence intensity. So by looking at the fluorescence, we can know which cells are active at what time. Okay. So let me move on to the next one. So first, analyzed the effect of TBG on the dendritic spines. So we implanted the cranial window at about P30 and waited for a month for the recovery. And then we image the dendrites once, and then subject the animal to 7-day stress. And then immediately after stress, we image it again and give the TBG injection, wait for 24 hours and re-image it. So since we can use the blood vessel patterns here to relocate the same dendrites across different imaging sessions, we can really pin down which dendritic spines persisted and which ones are lost or some new ones are formed. So as you can see here, these arrows point at spines that have been lost after these 7 days of stress. Right? This guy is gone. This guy is this guy. This one is gone. This one is gone. Right? This one is gone. And then there's some new formation over these 7 days, but it's relatively low. And then over that 1 day, the new formation is actually quite rare. I mean, in this particular case, I don't see any. These are like philopodia rather than spines. But with TBG treatment, over 1 day, you actually see a lot of these new formations. And interestingly, this one, for example, was this one, right? So a spine that was lost during the 7-day stress was restored by the TBG treatment. And this lower one seems to be the reformation of this spine that was previously lost. So if you count, what percentage of spines are formed over this 1 day, and see in the control animals, this formation rate was pretty low, right, about 2%. And the stress actually doesn't affect it. But with TBG treatment, the formation rate gets significantly elevated. The elimination rate over this 1 day actually remains the same for these 3 groups. But as I just mentioned, interestingly, in the UMS group, only a very small fraction of the previously lost spines got reinstated. But with TBG treatment, you actually restore about 20% of the previously lost spines. So having found that the TBG treatment would promote the dendritic spine recovery after stress, we went on to see whether such changes would have impact on the functional properties or the neuronal activity patterns. Because as we know, if you change the neural circuit connectivity, that would have an impact on their activity patterns. So first we did a mesoscopic calcium imaging. Yes?
Excuse me. Yeah, so can previous slide. So it's something, not this one. So a couple slide forward. Not forward. Yeah. So here. So is something to do with new spine formation? Is that right?
Yeah. This is a new spine. So this is the new spine formation, right?
So just the 1 day treatment.
Yeah, just one shot. Actually, we just gave the drug like immediately after imaging.
So it's
just, and then we wait 24 hours. Yeah.
So is there any study according to this drug? So this drug have some influence on protein synthesis? So
that's a good question, and I think it is very reasonable to assume that with elevated spine formation, there will be associated protein synthesis, right? We haven't specifically looked into its effect on protein synthesis. I mean, it could be some indirect effect, right? yeah, but I think it is very likely that there will be upregulation of some protein synthesis. It's just unclear yet, like what kind of molecular mechanism it is.
Yeah, got you.
Jude,
Yes.
can you hear me? Following Yichang's
Yes.
question, do you think this effect may be like circuit specific? This imaging is in the somatosensory cortex, is that right?
Yes, this is in the parallel.
So maybe this is for certain neural circuits, neural types, neuron only express, 5-HT receptors, or do you think this is maybe generalized mechanism for all the cortical circuits? Or
yeah, this is a very good question. Well, we're jumping ahead a lot. So so basically, the previous study by our collaborators group, they have done a lot of biochemical assay for the TBG's binding partner. And yes, they found that one of the receptors is the 5-HT2A. So it is possible that and also, I mean, let me just jump forward a lot. I think we have done this. So yeah, so we have shown that if you block the 5-HT2A activation by ketanserin, then all the behavior rescuing effects are gone. So 5-HT2A activation is required for its, at least its behavior effects. And I think it is reasonable to expect that it is also required for its rescuing effects on the spine level. And we know that layer 5 neurons, the layer 5 pyramidal neurons, do express 5-HT2A receptors. But some other types of neurons, like the inhibitory interneurons in the middle cortical layers, they were also shown to express 5-HT2A receptors. So, if the hypothesis is holds that the TBG induces spine formation via this 5-HT2A receptor activation, then sure, you would look for this effect in neurons that express the receptor. But at the same time, we need to be cautious about this, because the TBG is not exclusively a 5-HT2A agonist. So for example, it also has some affinity for the 5-HT2C receptors and other types of receptors, and like the SERT, the serotonin transporter, and also the monoamine oxidase. I think they are also their like targets. So yeah, it's a complicated question, and I think definitely it's worth looking into, like whether other cell types would respond to TBG similarly, or in a more cell type specific or circuit specific manner.
Yeah. Okay. Thank you, Drew.
Sure. So, okay. So let me go back here. Okay. So first, our functional study started with the mesoscopic calcium imaging. So we collaborated with Dr. James Ackman's lab, also at the UC Santa Cruz. So they built this mesoscope, which basically, you have camera lens to image rather, and you use a LED for illumination, and use a CMOS camera to capture this, basically the whole field view. This is one photon, LED illumination. And here, this LED basically provides infrared light with which we can track the behavior of the animal. So the animal is head fixed, but it can still like whisk at will. So when we take after we take the images, we have this analysis pipeline in which we first turn the raw images into delta F over F. And then we run through PCA or ICA pipelines, or independent component analysis, which decomposes these delta FOF images into different spatial temporal components. And we can throw out the components that correspond to blood vessels. And the remaining signals putatively coming from the neurons in there, although at a relatively low resolution, but we have a pretty big spatial coverage. And at the same time, because we have this infrared camera that is synchronized with the CMOS camera for calcium imaging, we also know at every moment whether the mouse is whisking or not, right? So we can analyze this behavior video by optic flow-based algorithm. So here you can see, these vectors have a larger magnitude, so that indicates like the mouse is whisking. And here you basically have very little optical flow vectors, so that's not whisking. And let me show you an example here. So these two are synchronized. So the mouse whisked, now it's not whisking. And let's wait, okay, it's whisking again, and you can see these bright signals emerging here, right? You can also see this negative image of the blood vessel, which has been subtracted. Okay, so so first of all, if we just integrate this signal and plot that against the magnitude of whisking over time, we can see like the calcium signal and the whisking are pretty well correlated. And in fact, if you compute the correlation coefficient in control animals, it's like above 0.4. But the UMS disrupts this correlation, right? The whisking and the calcium signals correlation dropped significantly. And after TBG treatment, this correlation is restored. And then if we look at individual episodes of whisking, so here each row is actually one whisking event, and the time 0 is the detected onset of whisking. So if we look at the mesoscopic calcium signal within this -2 seconds to +2 seconds window around the whisking onset, we can see in control animals, there's a pretty clear increase in the calcium activity. Right? It's well time locked to the onset of whisking. In the stressed animals, this time lock phenomena is like severely disrupted. You have all these activities like before the onset. But in TBG treated animals, the time lock is restored. And we can actually quantify this. So we define a whisking modulation index, which is basically the average calcium response in the 1 second after the whisking onset minus that in the 1 second before whisking onset. And here you can see in this measurement, the modulation index is significantly decreased after stress and restored by TBG. But interestingly, the response delay time, which is measured as the peak of the response after the onset, that time delay is not significantly changed. So this kind of suggests that the animal still responds to to whisking. It's just like in the non-whisking period, the brain somehow gets very noisy, right? So the modulation is decreased, and you may think of it as a kind of decreased signal to noise. And then after that, I move on to do 2-photon calcium imaging to achieve single cell resolution, to really analyze individual neurons' response to the whisking and to the behavioral tasks. So here is just another example of the population of the neurons we imaged in the primary somatosensory cortex, in the barrel cortex. And when you take this image, you can project it over time for each pixel and compute the standard deviation. So here, if there's no intensity change over time in the standard deviation image, it's going to be dim, right? Neurons that are more active will show up as brighter in the standard deviation image. So here you can clearly see the contour of individual neurons. And then we just manually circle the contour of the neurons and use that to extract the calcium transients over time for each neuron. And that is shown here as an example. So here we are just recording the spontaneous activity of these neurons in the stressed mouse. The mouse is head fixed, but it's not required to do any particular task. And you can see each row here is one neuron. And the red signal pixels indicate that is high delta F over F0. So that indicates like a high calcium response. So in the stressed animals, the spontaneous activities get higher than the control animals. And after TBG treatment, it's kind of restored to the control level. And here's the quantification. So first we can actually detect what counts as a transient, right? So basically we can compute what is the baseline level. There's of course going to be some noise, and we just use that noise band to calculate its standard deviation. And I define like a transient to be a peak that rises above that, that 2 times this noise standard deviation. And therefore we can define when is the onset of a transient, when is the offset of a transient. And the average transient size is just the area under the curve. So here, as you can see, the average transient gets bigger, but the transient frequency actually doesn't change. So it's essentially like the transients either get taller or get lasting longer in the UMS animals. And then we can also analyze the synchrony between different neurons. So here's an example to illustrate how we can do that. So the white traces are the DF over F traces. Here you can see the individual transients. And first I would just binarize these, right, in the red lines. So it doesn't matter how high the peak goes, right? As long as it is above this threshold, it is counted as as a transient, so it's 1. And for each neuron, we can do this binarization. Then we sum the responses of all the neurons point by point to get this red curve, right? That is the population response after the binarization. So basically each point will indicate like how many neurons are active at this time point. And then, of course, neurons could be firing completely randomly, but just by chance, some of them will be will be co-active. And in order to decide, what is the chance level of co-activity, I did a random permutation. So for each neuron, I would randomly do a circular permutation of this trace, the red trace, and then sum them up to generate a new surrogate red trace for the population response, and repeat this process many, many times. And then you can take the average population response and get the 95th percentile value. So that is this yellow dotted line. So above this line, we can take these peaks as population synchronous events. That is, beyond this random coincident firing. And if we look at these synchronous events, we found that the average duration of synchrony is increased in the stressed animals. The percentage of time the neural circuit is in synchrony is also increased. And the percent of neurons that participate in each synchronous event is also increased. Right? And TBG basically normalized both, all 3 of these metrics. So far I've been, characterizing all these brain activities in just baseline conditions. The mouse is not engaged in a particular behavioral task. Next, I would like to examine the behavioral responses of the animal. And there we used this head fixed paradigm of the texture discrimination task. So we put the mouse on this neurotar device. So you can see these little holes. They can actually puff out air to lift this black arena, which is very light. It's made of a carbon fiber. So the mouse can actually run on this arena, and because the mouse is head fixed, the arena would actually move in the opposite direction, right? So equivalent, the mouse can actually explore different corners of the arena. And here's just the example of the neuronal firing as the mouse moves in the arena. And on the arena wall, we can have like different textures of sandpapers attached. The mouse can still explore them. Right. And the test here is analogous to the free moving version. But first, we would get the mouse to, in the test, we get it to run for 5 minutes. And then that's the encoding phase, right? Exploring the same texture. After 10 minutes rest, we change the textures, and the mouse is subjected to this testing phase with a novel and a familiar texture. And we just use the program called Boris to annotate the mouse behavior during both encoding and testing phases. So we can annotate when the mouse is running, when it is whisking, and whether it is on the left texture or right texture during the encoding phase, or on the familiar or novel textures on the testing phase. And when we quantify the mouse behavior parameters, we found that in all three groups, the total amount of interaction time is not that significantly different. But again, the control animals exhibited a preference for the novel texture, which is abolished in the stressed animal, and the TBG restored it. So basically, the behavior response in this head fixed version of the texture discrimination task is the same as in the free moving version. But here we can actually do simultaneous calcium imaging on that, right? We would first do the virus injection and window implantation P30. And a week before our stress starts, we would habituate the mouse to this neurotar device, so the mouse doesn't get freaked out on the head fixation. And then it's the 7-day stress, and after the stress, we give the TBG treatment, and then after 1 day, we do the behavior plus imaging. And here's an example of what we see with the calcium activities, with and without a texture interaction. So here, these purple lines indicate the time when the mouse is interacting with the novel texture, and this kind of bluish gray color indicates when it's interacting with the familiar texture. So this is just a small sample of the neurons that we can image from a single mouse. And so first of all, we found that if we compute the average DF over F of during interaction with a texture versus non-interaction, during interaction, the response is much higher, right? And it is not significantly different among the three groups. But when the mouse is not interacting with the texture, UMS animals actually exhibit a higher average DF over FCL. So that is actually in line with the finding in these baseline conditions, right? That after UMS, somehow the baseline activity gets elevated. And then we can really look into individual neurons' response to the two types of textures. So for each texture interaction, we would take the 1 second, actually starting from 0.3 seconds to 1.3 seconds. we choose this window based on previous publication from Fritjof Hamilton's group. I think the idea is like you probably want to avoid that the initial contact there may be some like mechanical impact, and that can potentially cause some artifacts. So they use the slightly later time window, and we followed their definition. So that's the window of response. And then 1 second before the touch onset, we consider that as the baseline. So now we can then define this touch response as the average DF over F over this response window minus the average DF over F over the baseline window. And in control animals, we found that when the mouse is interacting with the novel texture, this touch response is much, much higher than the response during interaction with the familiar texture. And such a heightened interaction response to novel texture is abolished by stress, but partially restored by TBG. There's still significant difference, although the absolute response magnitude is not as high as in the control animals. And then for each neuron, we can actually determine whether it has a preference to respond to the novel texture or to the familiar texture, or to both. So here I used this so-called receiver operating characteristic, or ROC analysis. So basically so first we do the novel texture. We would take all the interaction with the novel textures, and we take the response and we take the baseline. And for the response period, let's say we have 10 interactions with the novel texture. So we have 10 values for the response period and 10 values for the baseline period, right? And the distribution of these two sets of values may overlap. Sometimes the neuron may even respond more, be more active during the baseline than during the interaction. So the ROC actually determines, for some optimal threshold, like what is the best you can do to differentiate between these two distributions of values. And if the two sets of distribution are very far apart with only a little bit overlap, then you can be very, very confident, right? When you see a particular response value, you're very confident whether it is during the interaction with a novel texture or during baseline, right? And in that case, if a neuron exhibits that kind of response pattern, we call it a novel selective. Conversely, during the interaction with the familiar texture, if a neuron exhibits much elevated response during the interaction compared to the pre-interaction baseline, then we consider that as a familiar selective neuron. And some neurons are actually responding to both, right? So they are non-selective, as shown here. And there are also neurons that are just not really responding to texture interaction. So so when we classify neurons based on these criteria, we found that in the control animals, about a third of the neurons are novel texture selective, 6% are familiar texture selective, about 9% are non-selective, and the rest are non-responsive. After UMS, the great majority of neurons become non-responsive. I mean, here, non-responsive does not mean like they are not firing at all. It just means like in the baseline versus the interaction period, there's no significant difference to differentiate the two. And after TBG, the novelty response, the novel selective neurons get much restored. So are the familiar selective neurons. And if we compute how many neurons respond to the novel texture, and by the way, here the definition is the novel selective plus the non-selective, right? Because these neurons also respond to novel texture. So in control animals, it's about 40%. In the UMS, it's like practically abolished. And then TBG like significantly restored this population. And interestingly, if we look at the familiar texture, remember the first time the animal sees this familiar texture is during encoding, right? And the first time when it sees that, it's still novel by definition. So we found that neurons that respond to this later familiar texture during the initial encoding phase is actually pretty high in all three conditions. And during testing phase, all of them exhibited this significant decline in the percentage of neurons that still respond. Right? So that suggests that the animals, even after UMS, they still know what is novel and what is familiar. The problem that we see here, like there are very few neurons responding to the novel texture, like we call it a novel texture, is probably because like the animal's sensation is blunted by stress. So it is interacting with the texture, but it can't really differentiate between the novel and the familiar textures. So it somehow just treated it as if the novel texture was also familiar, and therefore, the response was suppressed. But in other words, there is this ability to detect the texture, and there is this ability to decide, okay, this is novel or this is familiar, and make correct behavior. It seems like the preference for novelty per se is still there after stress. It is the sensation of the particular texture that gets impaired by the stress. So in summary, we found that stress induces affective, sensory, and cognitive deficits in mouse. And in the brain, it leads to cortical dendritic spine loss and disrupts the task-associated activity patterns of the cortical neurons. And with this novel non-hallucinogenic psychedelic analog TBG, we can rescue these both the behavior and the neurobiological deficits. And such rescuing effects basically tells us there's great potential in psychedelic analogs as treatment for stress-related mental disorders. And I would like to thank my advisor, Dr. Izzo, and the members in the lab, in particular, Dr. Michelle Tija, who's now a postdoc at Stanford, and she really initiated this project and did a lot of the behavior and the calcium imaging experiments. Dr. David Olsen's lab at UC Davis supplied the TBG, and they really introduced us to this psychedelic field. And Dr. James Ackman's lab at UCSC and performed the mesoscopic imaging with us. And Dr. Lu Chen's lab at Stanford actually did some slice electrophysiology experiments for this study, which I didn't talk about today, but if you're interested, I can talk about it during the Q&A session. So before I end, I actually would like to take this opportunity to make a little bit of advertisement for a small side project. So this was an effort by Dr. Izzo and me, and also Talia Lieberman, who's a very talented art professional in San Francisco. So we set up this website, artneuroscience.com, to try to explore the sort of similarities as well as the contrasts between visual art and neuroscience. We are putting in some projects at a relatively slow pace. And you're welcome to visit this website. And if you have any interesting ideas, either about art or about neuroscience, please email us, and preferably, if you have any beautiful scientific pictures to share, that will be really great. So I think I will stop here, and thank you again for your attention. And if you have any questions, I'm more than happy to discuss. Thank you.
Sure, I think there are a few questions in the chat box.
Sure. Okay, so let me see the chat. What is the TBG injection dose in mouse? So we did 10 milligram per kilogram. Have you ever tried to administer TBG before the stress event? Yeah, that's a very interesting question. No, we haven't. I think it's definitely worth doing to see if it is protective. Yeah, we haven't done that, but that's worth trying. Any other questions?
Hi, I remember in the literature you show us stress, they can even decrease the number of dendritic spine, dendritic branches, right?
Yes, there will be some, the dendritic branch tip, those high order branches, some of them may retract. Yeah, I didn't focus on those because the spine dynamics on those like terminal branches seem a little different from those on the branches that are not at the terminal. So I focused on the non-terminal branches here.
Yeah, I just don't know whether 7 days stress is long enough and severe enough to affect the dendritic branches.
Yes. So I have to check that, but in the previous work, also from our lab, so in that 7-day restraint stress actually does cause a dendritic tip retraction. So can you, am I still sharing this? Oops, give me a second. I think there's some. Yeah, so this was from the previous paper by Dr. Eric Chen in the lab. So he showed that after 7-day restraint stress, there is indeed a dendritic tip retraction. So yeah, as I said, I haven't checked this in the in my stress paradigm.
Yeah, thank you. And the other question is, I remember all of your statistics. The TBS, they can almost rescue to the same level of the control group, except one, except the first day formation of the spine. In that panel, the TBS group, they can recover even higher than the control group.
You mean this panel?
I couldn't say it. I couldn't.
Oh, you couldn't see it?
Yeah,
oh, sorry. Just now you so sorry. I thought I was now can you see it now? Is this this panel?
Yeah, I mean this.
Yeah. So here actually, formation over 1 day is not really affected post the UMS, right? Because here these show like over this 1 day, that is like after UMS has concluded. So the formation rate is not changed, but TBG really boosts the formation. And we believe that this sort of partially compensates for the loss of spines during these 7 days. And let me show you this, which I didn't put in here. So let me see. There is here. So here it's like over the 7 days of stress, right? So we image before stress starts and image again after stress ends. So over the 7 days, the UMS doesn't affect the cumulative formation over 7 days, but it elevates significantly the elimination, right? So you have more spines lost during that 7 days of stress. And then after stress ends, you have the part of those lost spines restored by TBG. Right, as shown here, it's 20%, right? So there are still a lot of spines lost that are not restored, but it is significantly higher than what you would spontaneously recover after UMS.
G.
Yes.
Hi.
Oh, hi.
Hi. It seems like the TBG just rescued all the phenotypes that are caused by UMS at all levels. So my question is simple: is there anything that this drug doesn't rescue?
I mean, it's well, I mean, so certainly, what we did is not exhaustive of all the possible effects of stress. I mean, one interesting thing that I'm trying to do is to see how long the effect would last. Right?
Mm-hmm.
Yeah, you would like it to be long lasting. And also, if you look at this, like some of the rescues are not complete here, right? Like in terms of the responses here, I think you probably, I mean, it's probably not significant, but there is a trend, right? It's, so yeah, I mean, we don't know whether this is the best regimen for treatment of stress. We tried to go for a relatively low dose, right? Because we want, I mean, it seems like desirable if you could do with a low dose, and we only did one single dose. So yeah, I mean, it would be necessary to better characterize the optimal regimen and to achieve like a better therapeutic effects.
Yeah, thanks.
So yeah, I have a couple questions for you. I'm Yi Chang from Tsinghua. So the sensory input could be from visual, from olfactory, or from the gentle touch. So is that something that I heard that you said lower object recognition not different between something? I'm not very clear.
Yeah. So I mean, I didn't do the novel object recognition test in this stress paradigm, but a previous study using the 7-day restraint stress suggests that the novel object recognition test, I think that was using visual cues, it was not affected.
So the question follow-up is the calcium signaling. So in the, for example, the visual cortex. So is that different from the sensory cortex? Is there something that TBG cannot rescue or something like that?
I'm so you're thinking like why stress does not affect the visual system as much? Is that the question?
So the question is the specificity for this particular paradigm. So it looks like the animal is more sensitized to the gentle touch than the other cues. So
I see. Yeah. So I mean, I can only speculate because the mouse is not really let me put it this way: the mouse doesn't have a very high visual acuity, right? So behaviorally, I think it relies much more on the like olfaction and the whiskers to probe the world than its vision. So maybe that means the visual system doesn't have that high sensitivity, and therefore at the same time it is like more robust. And given that we were only doing like 7 days stress, probably it's not sufficiently strong to disrupt like visual perception yet. I mean, that was just my guess. And also, you know, the two systems would receive like different inputs, right? So it is also possible that you know, the different input regions to visual versus somatosensory areas are, you know, are sensitive to these stress manipulations to different extent, and that can also lead to this, you know, different robustness of the two sensory cortices to stress.
So in your last slide, you said something that the animal still can distinguish the lower objects. That means so they can differentiate the object, but they cannot sense the text. Is that what you mean? Yeah.
Yeah. So here, I mean, okay, let me. That's a little. Let me try to rephrase this. So here you see this is the same texture, right, presented to the animal twice. The first time the animal interacts with this texture, it elicits a relatively high response. And these three like blue bars, they are not significantly different. So the animal is responding to this texture about the same. You know, there's a trend for UMS animals to respond a little less, but you know, roughly the same. And then the second time it engages with this same texture, the response significantly decreases, right? Now, so I interpret this to indicate that the animal actually knows, okay, I am interacting with a previously encountered texture, right? And that also persists in all three groups. So you always have this significant decrease in the response when the texture is presented to the animal in the second phase, in the testing phase, right? But then how do we understand the reason why the true novel texture, when it was presented in the testing phase, it failed to elicit the high response here? And my thought was that the animal just knows, okay, you know, the first time the animal interacts with the texture, it just knows, okay, this is something new. I've never interacted with this. But it can't really tell whether it is a 60 grits sandpaper or a 200 grits sandpaper. So the second time when you present it with the novel texture, it only knows, okay, I'm encountering with this sandpaper again. It can't really tell the subtle differences. So it just treated it as familiarized texture, and hence this greatly reduced response. So that's what I mean, like the animal still knows this preference for novelty, but it fails to distinguish between the two textures.
这个地方可能我还是没 get it， 就是我不是知道，我不清楚大家有没有 get it， 就我还是有点 confusing。我的 understanding 好像是你在说，好像是就是蓝颜色的 bar 是一个 encoding， 这个是一个一个物体是吧？也可以把它变成一个物体，也可以把它变成一个 texture。就这时候在蓝颜色和红颜色之间，你有没有 change texture？ 在这个 moment？
Here, no, the texture is not changed here. This is the same texture.
So still, the animal still can differentiate. Differentiate 其实是 lower object 是吧 ？lower object 它可以分，是吧？等一等，这边再等一等，就是说 就是说这边的蓝颜色的 bar 和红颜色的 bar， 它的 object 其实是不一样的 ，shape 不一样吗？什么地方不一样，它可以分离开来？
No, here it is the same bar. It's just like during encoding, that's the first time, right? The first phase. So,
Okay.
so it's here. The blue bar is during this encoding phase. The red bar is during the testing phase. But it is the same bar, right? It is the same bar, just presented twice, or the same texture, presented twice. Yeah. So
这个地方如果 shape 是一样，所有的东西都是一样，只是 texture 不一样，它是分离不出来的，是吧？你在 test 的时候它是分离不开的，是这意思吗？
Yes, that's what I guess. So in the stressed animal, when you during the testing, you actually give it two textures, right? One is still the same. So one is still the same. We call it familiar because that was there before, and the other one is the novel one, right? So in the control animal, it should be able to tell which one is which, right? But in the stressed animal, it seems like it can't tell. It was just probably thinking this was identical to the familiar one, and therefore it showed no preference. Is that clear or?
就是你在 encoding 的时候，你放的是 same texture， 它们的 shape， 它们所有东西都一样的，是吧？
Yes.
一样的。然后你这时候在 testing 的时候，你 change 一个 lower 一个 familiar。
Yes.
这个在这个experiment 里面，你 change 的是肯定要 change something 了 ，something 肯定要 change。你这时候 change 的是什么？
我 changed
的是
，Yeah, I changed the coarseness of the sandpaper. 就把这个本来是同样的这个砂纸的两边贴着，然后我后面换的就是我把它换成那个，你不是砂纸可以买，你有的细有的粗的嘛，我就给它换一个不同的粗细。
那刚才那个experiment 里面，你说它还是可以 distinguish， 就最后一张 slide， 这个地方是怎么做的呢？你在
Okay, so this is actually the same. I mean, it's the same experiment. I'm just focusing on the response to this. I'm comparing the response during encoding and the response during the testing, but both to the familiar one. So it's still taken from the same experiment. Yeah, I can just isolate the trials in which the animal interacted with these, let's call it just cyan texture. 其实就是说我这第一次，它是同一个 texture， 比如说它这个都是我随便说一个，比如60，比如都是60这个grids， 那就第一次的 encoding 的时候，它的 response 高，然后同样的这个 texture 到 testing 的阶段的时候，它对这个 texture response 它就低了。
这个对的，就是说你对于同样的 familiar 低了以后，你不是对于那个 lower 的就高了吗？那么这样子就是应该还是说这两个可能是没有差别的。
对，在正常的情况下，对 familiar 的低了，对 novel 的就还应该这么高，对吧？就是说你因为第一次看到这个所谓的 familiar， 就是在 encoding 的时候，我们后来叫的 familiar， 它其实第一次看到它也是 novel 的嘛，因为它从来之前没见过，所以它第一次看到的还那么高。但我们的这个 novel， 你看这个是我们就真正的 novel， 对吧？只有在第二次才见，第一次没见到的，在这个第二次就是说见到这个 texture 的时候，它的 response 和这个 response 你看实际上是差不多的，就是说你上来如果真的是个新的。因为你这第一次看到它永远肯定是新的，你这就在第二轮的时候看到这个它确实也是个新的，但是 在这个 stress 的 animal 里面，他就不觉得这是新的，他只是觉得 OK， 我这反正又是个 texture， 那就是 texture 吧。就他不知道
这个地方有点问题，就是我不太 understand 的地方。如果是 UMS， 他在 testing 的时候，他如果是对于familiar 它还是下降了，对于 lower 肯定是上升了。就反过来讲 ，lower 的东西肯定是上升。
但是现在的问题是这样，假如说这些老鼠根本不知道这还有个 novel， 因为给 familiar 就是说在 encoding 的时候，它也是有2个，但 familiar 就是同时我有2个 copy 在里头。就你看，它这还是一左一右，有2个，只不过它们俩是同样的 texture。然后你假如说这个 animal 根本它 sense 不出来这个 novel 的，它就觉得我跟这个前面是一样的，还是2个一样的 texture。
那它应该这时候放的时间应该 familiar and lower 应该一样的，就不会在 familiar 下降了，不会在 familiar 下降，这个是 point。
是的，不，这个 familiar 下降是指的它跟之前的这个比，就是说不是它时间的下降，而是它对它 response 的 neuron 的这个 percentage， 就是说对 specific
这个地方是 neuron 的 percentage，
对，这是 percentage of neurons， 对，这个他们2个的时间就是 interact 的时间的话，那确实就一样了。在 discrimination 你看这2个就没有差了嘛，因为这是说我拿去 familiar 的次数减去不是去 novel 的减去 familiar 的这个时间，然后除以他们俩总时间，你看这些都接近0，那就说明它没有 preference。
这地方有点 controversial， 就是我是觉得好像是它还能分出来这个 contact， 它还能分出来 tactual。但好像他就是好像对于 lower 的这个 texture 好像不是那么care， 大概这个意思，是这个意思吗？
对，因为我的这个inclined to interpret maybe a little differently, because if the animal doesn't care about the novel texture, right? It doesn't care about novelty, let's say.
Yes.
If it doesn't care about novelty, then if you look at this, there shouldn't be this many neurons responding to the texture during the encoding phase. Right, because during encoding phase, whatever you give it, it's new, right? It has not encountered this before. Yeah, so let me see. Duration of yeah. 不好意思，要不我先这个可以回答一下别的问题，然后咱们可以接着再讨论，好吗？
对，可以，没问题。我只是说我这边有一个小问题。OK， 拜拜。没有，你们继续，你继续。
So can ketanserin inhibit TBG synapse formation? Yes. You mentioned the duration of effect. Do you mean timing? Yeah, yes. So do you know the TBG effect is persistent after multiple rounds of stress? That we haven't done. So we only stress the animal for 1 round. So that question is essentially like if you give TBG and then give it a stress again, like whether it can somehow prevent the second round of stress from damaging the brain. Now that we haven't done yet, we don't know. It would be nice if it can prevent that.
Sorry, Zhu, just to clarify here, the second part of my question, I was just wondering if you can, see the same effect after, multiple rounds of stress. I mean, will the TBG remain effective after multiple rounds, or does the effect, wear off? Does the animal essentially get habituated to the effect of the drug after the first round or two?
So let me clarify a little bit. So here the TBG, we only gave the animal 1 dose of TBG, right? So I mean, your question, like what is the scenario that you are thinking about. So you are thinking about we first like stressed animal for 7 days.
Well, the
Then
scenario, the scenario that I'm thinking about, I mean, I think what you found is really interesting, and I'm just wondering, you know, it seems like it's, you know, got some pretty interesting effects that will help ameliorate the effects of stress. And I'm just wondering, you know, how many times can you get that same effect on stress? I mean, does the animal, does the effect of the drug kind of wear off after you give it once, or do you have to give it more the second time, or will it remain as effective, you know, over time? Essentially.
I see. So I mean, please let me know if I understand you correctly. You are imagining a scenario like you stress the animal, then give TBG, and the animal is rescued. Then after some time, you stress it again, give the same dose of TBG, see the animal, if the animal is rescued, then stress again. So let's see, because stress can like repeat, right? And then see if eventually at some point the TBG no longer is effective. Or
yeah, that's exactly what I'm curious about.
Is that yours? Oh, I see.
Yep.
Oh, that's interesting. Yeah. No, we don't know yet. We don't know. We haven't done that yet. Yeah. Yeah, that's actually a very interesting question, because that's going to be very relevant to clinical medicine, right?
Sure. Yeah. All right. Well, I'll stay posted. Thank you for the very interesting talk.
Thank you. So Jin Z will be the next one to ask.
Yeah, I'm Jin.
Okay.
So a very interesting talk. I missed some parts, so I have a couple questions for you. So first one is how you induce the stress to the mice. So what kind of model you are using to induce stress?
Oh, okay. So if you missed that, so we used this so-called unpredictable mild stress. So basically, we have different stressors, like, you know, change the light exposure, like we turn on the light at night, or, you know, put it in a tilted cage, or restrain food for overnight, or like put it into a Falcon tube for restraint, like, and you know, either social isolation, or put it into a group of stranger animals, or sudden loud noise, or island isolation. You know, you put the animals on little islands surrounded by water, and mouse really don't like to be in water, so they're kind of you know, freaking out standing on those little islands made by Lego blocks.
So the poor mice on the 7-day stress. So I got
it. So you give them sort of quite a randomized stressors, right? And yeah, we believe that is kind of, you know, that's going to mimic this randomness and unpredictability of stress in real life.
So do you know which pathway you are affecting for the stress? So
you mean by pathway, you mean the like a brain circuit?
What kind of pathway or what kind of receptors was affected in the stress model?
Oh, you mean the like the molecular pathways?
Yes.
No, we didn't look into that because, you know, we're not a molecular biology lab. We are, you know, technically not equipped to do that kind of investigations.
But why you use a drug? Because you are using drugs to treat the stress. So
okay. So so the reason that we used this drug TBG is because there are a few things we already know. So first is that in some previous studies, and including some previous paper from our lab, it shows that stress can really cause disruption to the synaptic circuit in the cortex, and specifically in the somatosensory cortex as well. Here in other groups that showed that it also disrupts the synaptic circuit in the prefrontal cortex. In the cortex, you basically, after stress, you see this reduction in the dendritic spines. And as you know, spines are the post-synaptic site of synapses, right? So if you lose spines, that means you lose some synaptic connections. And if you lose synapses, you can have the connectivity basically of the network is disrupted, and that can lead to all sorts of abnormal neural activities. So that's the target that we tried to correct. And the reason that we tested this particular drug is that because in the previous paper published by our collaborators, in which our lab also contributed a bit, we found that this drug actually promotes dendritic spine formation. So that is the motivation that we believe that this drug may be able to somehow repair this disrupted synaptic circuit.
But they don't know how the drug increase the dendrite formation is through which pathway. They don't know which pathway.
No, that is that is the to be studied. I mean, we only know that this drug is also a 5-HT2A receptor agonist. Okay. So they did some biochemical assays, and they also used blocker of the 5-HT2A receptor, and showed that if you give the blocker the ketanserin, you can actually block the response, the rescuing effect of TBG. And in their paper, they also showed that like if you add ketanserin, you can actually block this pro-spine formation effect of TBG. So that is involved. But as I said earlier, it is not a proof that this is the exclusive pathway through which TBG exerts its rescuing effect. So it seems like the molecular signaling is like quite complicated here.
Yeah, because you also mentioned about the NMDA receptor, the reduced NMDA receptor also correlate with your stress phenotype.
No, we actually didn't do that experiment, but that was in some previous studies, people show like there's a reduction in the NMDA and AMPA receptors. Yeah, so it's it's actually a very interesting and important question, like what is the molecular signaling pathways that are involved. But that needs a lot more studies.
Yeah, because why I'm interested in this is I'm studying the NMDA receptor. So we got some patients, we cloned some NMDA receptor antibodies from the patients. So I gave the antibodies to the somatosensory cortex and then reduce the anxiety phenotype in the mice. So I just curious about that because I'm not studying anxiety before, but I see the phenotypes in the behavior phenotype. So I just wonder how the NMDA receptor involved in this kind of stress pathway. And you mentioned about the somatosensory cortex actually very important for the stress, maybe is key for the stress modulator or something. Do you have a clear pathway how this involved in how the somatosensory cortex involved in the stress pathway?
Well, as you know, classically when people talk about stress responses, people would focus on the limbic system, right, or the prefrontal cortex, or hippocampus. And I mean, the sensory cortex is like it's relatively less studied compared to these other brain areas. I'm not saying like the sensory cortex is somehow a generator of the more classical sort of stress-induced behavior deficits. I'm just thinking like, first of all, we actually probe the sensory defects a lot less, right, in the context of stress. And there seem to be some clinical evidence, some more like anecdotal reports from doctors suggesting that in psychiatric disorders, sort of in general, often there are some sensory defects. Those are probably a bit subtle, and the patients usually do not focus on those, right? I mean, certainly like a clinical diagnosis criteria are not focusing on these sensory deficits in the context of like psychiatric disorders, but they probably are there, and one may even argue that certain sensory deficits can have effects on the downstream circuits right? Because that's where you get the stimuli from the environment. First, you have to go through the sensory areas, and then that information is relayed to higher order brain regions for like further processing and decision making and so on. So that's why we think it's probably worthwhile to look into the deficits in the sensory cortices. I mean that's not to say like that would somehow supersede these other brain regions in their contribution to stress. It's just a different aspect of the damages that the stress can do to the brain.
Just I'm curious, do you know any study they have traced from S1, so which is upstream or downstream to S1 in the stress pathway? Do you possibly know this?
You mean like what brain regions?
Yeah, like
of
prefrontal
S1
cortex. So how the prefrontal cortex, like you mentioned about prefrontal cortex,
yes.
and the VTA. So what's the relationship between these different cortex to the S1? So is there a cascade for the stress pathway to?
I mean, S1 would primarily, they would project to what? Like they project to the motor, they project to contralateral S1. They probably project back to some high order thalamic nucleus. They project to S2. Actually, I mean, for me, I'm not so familiar with the direct link between S1 and I think PFC probably receives some S1 input as well, but I'm not familiar with any evidence that there's a direct sort of causal link between the deficits in S1 and in these other brain regions. Yeah, but I mean, I think it's imaginable that if you have some problems with S1 and that causes defective sensory processing. So the information it sends to the higher order regions is going to be distorted, right? And that can, in fact, lead to downstream problems because you are feeding in some erroneous information. But I think it is a little difficult to establish a very clear-cut sort of causal relationship because, I mean, stress is given to the animal behaviorally and therefore it is systemic, right? And the effect shows up sort of at the same, I mean, it's hard to say what is the effect of stress on S1 and then from S1 to these other areas versus effect directly on the areas downstream of S1 anatomically.
So
yeah, I mean,
so from your evidence, you know that S1 is very important for the stress, the happening.
Well,
can we move on to other people's questions?
Yeah, maybe let's we can talk about that.
Can we talk later?
Yeah. So let me see. So you have so how to identify the good modulation to spine number. So could you please clarify this question a little bit? Like what kind of modulation to spine number? What are you trying to quantify here? Hello?
Not sure.
Sorry, I can't hear you.
I mean, can you unmute yourself, ask the question again?
So, okay. So I think you asked like how to identify the good modulation to spine number. I mean, could you please clarify the question a little bit?
I'm trying to unmute this person, but he.
Oh, so maybe let me type. Let me type hi. let me try to type. Could you please clarify your question a little bit? By modulation. Oh, how the reduced spine number contribute to the drug effect? Oh, okay. So here's the thing. Let me first show you this one. So the question is like how does reduce the spine number contribute to the effect? So first, this I didn't show in the main slide, but basically, if you look at the number of spines on these apical dendrites over the 7 days of stress, the formation, the number of new spines formed during this period is not changed, but the number of spines eliminated gets increased, right? So the circuit is actually losing some connections over the 7 days of stress, right? Now, when you give the drug over that 1 day, right? So previously we are talking about the changes over this period of time. Now over this 1 day, after you give the drug, it doesn't really change the spine elimination over 1 day, but it boosts up the formation of new spines from P67 to P68, right? And also you have some of the new spines that are formed at the exactly same place where previously a spine is lost. So this percentage really sort of restores the part of the lost spines. I mean, of course, it is not a complete restoration, but we believe that this kind of at least is correlated with the behavioral rescue, right? And conceivably, it contributes to the reparation of the synaptic circuit. Does that answer your question?
Yeah, but I want to know that why the animal have to reduce some spine number, and how to. Okay.
So go ahead.
I wonder where is the threshold of it have to reduce the spine number, and how much spine, how many spines it should remain to remember the test.
Oh, so why do they have to reduce that? I mean, the reduction in spine numbers is actually a kind of a pathological outcome of stress, right here.
Yeah.
Are you asking like why even in the control animals there's this elimination? Is that the question?
Yeah, but I mean that the 5% may be the normal threshold, but why it only remains 5% will could make the animal remember the task.
No, I'm here. It's not like well. I mean, so first of all, there is a continuous formation and elimination of spines, even in a control animal, without any training, without any intervention or perturbation. So it is just a natural sort of process of a turnover. And as you can see, in at this age, right, you have a relatively constant number of spines. So the number of new spines formed over a period of time would roughly be equal to the number of spines eliminated during that period of time. Okay, so that is just a baseline turnover. I have to say it is not clear yet why there should even be this baseline turnover. personally, I think it is just a way for the system to remain sort of plastic so that it can have the potential to reconfigure itself when there is some new situations arising. And it can, reconfigure the neural circuit to maybe store new information or to perform some tasks better and so on. Yeah, so that's the baseline turnover. And then with the stress, you actually lose too many spines, I would say, right? You have this significantly elevated elimination. So you are actually disrupting the circuit. And that's why we think that this, elevated formation, you actually grow more, so compensates that. But it's not saying like, specifically these 2% of newly formed spines are encoding some particular tasks. No, these animals were, either just stressed or, stressed plus like TBG treated. They are not trained on anything and they are not tested on any behavior during this period. So they're just experiencing whatever a normal animal would experience in the cage, besides like they are stressed, right? So it's not like we are training it to remember something. I don't know if that answers your question or. Hello, I can't hear you again. Hello? Hello?
I think maybe because it's internet.
Yeah. Sorry. I can't hear. And let me see how is there. Somehow I can't even see the. Oh, now I can see the chat. Okay. Oh, okay. All right. So is there any other questions? Okay, you can hear, but apparently we can't hear you. Yeah, but somehow we can't hear you.
Any more questions for Ju? In that case, I think we will stop recording, and thank you, Ju, very much. And then I think also Jing and Jia Shizhong has more questions for Ju. So
okay, yeah, so we can stay here and talk more. So you will stop recording, right?
Yeah, it was stopped.